All right, why don't we start with a sound check. Can you hear me in the back there? Can you hear me in the back there? Okay, great. I apologize for being late. A student staggered into my office about 15 minutes ago on the point of collapse. I didn't think any of you would want me to rush back over here till that was straightened out. She's doing much better, but that's why I'm late, and I apologize. Any questions about Wednesday's lecture before we continue? So, wakey, wakey. I think I'd like to just go over one more time the Z scheme and the cyclic electron transfer, just because it's somewhat cosmic, and to make sure that you're oriented before we head into the dark reactions. What would that be? That would be 18. But I don't know why it's so slow. The panel that I'm trying to open is number 18, if you want to get there ahead of me. There we go. And so the point I'd like to make with this diagram, if it opens up properly, there we go, is that in the light reactions of photosynthesis, as in the electron transfer reactions of oxidative phosphorylation, you have a series of membrane complexes that are capable of transferring electrons between various electron carriers. And so you have a very bad user interface. You have photosystem 2 that's embedded in the membrane that's going to absorb light and generate a highly energized electron. It will transfer to in the membrane electron carrier of plastoquinone. Its electrons will go to a membrane complex that spans the membrane, cytochrome B5F, B6F. Its electrons will go to plastocyanin, small peripheral membrane protein, analogous to cytochrome C, although not identical, which will then transfer electrons to photosystem 1. And photosystem 1 can either transfer its electrons to ferredoxin and then to NAD+, which would be available, or it can go back and cycle around through photosystem 1 repeatedly. And the point of all this is that photosystem 2 and B6F are both membrane complexes that can generate a proton gradient the way the electron transport generated a proton gradient. And we talked about the ATP synthase that has a great deal of structural homology to the same kind of ATP synthase in the mitochondrion. So I think we'd gotten started on rubisco and what that wonderful molecule did. And I think we went through the intermediates, did we, of the enzyme reaction of rubisco itself. Is that correct? Okay. I can trust a safe answer to that because no one wants to repeat enzyme mechanisms unless they have to. Okay. So we talked about how rubisco could pull off this truly amazing trick of adding a carbon dioxide to a 5-carbon molecule, ribulose 1,5-bisphosphate, to make 2 glyceraldehyde 3-phosphates. But it's a very tricky reaction, and it's hard for it to be very specific. And unfortunately, rubisco can also slip and use oxygen as a substrate. And it would look something like this. When rubisco is making a mistake about it all, it can take the 5-carbon ribulose bisphosphate, 1,5-bisphosphate, and in this case, when oxygen is added, now you split the molecule into 2-carbon and a 3-carbon piece. This piece is fine, but the 2-carbon piece is something called phosphoglycolate. just a 2-carbon little bitty thing. It's got a carboxylic acid at one end and then the phosphate on that end. This is not particularly useful. And so the cell is going to have to use both ATP and NADPH to recover the carbons that are in the phosphoglycolate. And this process is called photorespiration. Photorespiration is not a good thing. It's a waste of energy. Oxidative phosphorylation is good. That's mitochondrial and stuff. Respiration by itself is usually referred to oxidative phosphorylation. But photorespiration is in the mitochondria, and it's a wasteful process. It's a byproduct. And the reason it's wasteful is because in order to get this thing back, this is what you have to do. No, sorry, this is not it. We can go over the chemistry if you like of what it takes to get the phosphoglycolate back, but that's not what this is about. So let's assume that the Rubisco is doing the right thing and is taking the 5-carbon sugar and making 2 3-carbon pieces for us. How, in the long run, does that actually help us make total net glucose, 6-carbon sugar? It's not as simple as just taking 2 glyceraldehyde 3-phosphates and smushing them together. Nothing is ever that simple in biochemistry. Instead, what we're going to take advantage of is something called the Calvin cycle. And it's going to kind of be switching 2 and 3-carbon pieces back and forth to rearrange the carbon backbones so that overall, over a period of time, carbon dioxide will be accumulated into bigger and bigger pieces. The Calvin cycle occurs in the stroma of the chloroplast, the kind of meaty inner stuff where a lot of other metabolism is going on. And it has kind of a funny story behind it of how it was discovered. Back in World War II, they just started making radioisotopes pretty much on demand, at least some radioisotopes. And up until this point, it had been very hard to study what happened when chloroplasts fixed carbon dioxide. There was no way to get at it. And so the hope was that with radioactive carbon dioxide, you could see where that was going. You'd mix in your radioactive carbon dioxide and flash the light and see where it went. In World War II, they didn't have a lot of sophistication about what they could make. And the isotope that they had was C13, which you don't hear about much because its half-life is something like 24 hours or something, maybe even 12 hours. It's a very short half-life. And in order to use it for their experiments, it was made up in the cyclotron on the top of the hill behind the University of California at Berkeley. And a very brave and hardy graduate student would get on a bicycle and ride down the hill, which if you've seen Berkeley is a very steep hill, to Calvin's lab where they would use it for the experiment right away because it was decaying the whole time they were riding the bicycle down the hill. And that's how the first insights into the Calvin cycle got discovered. So the reactions we need to look at here go something like this. You can start anywhere, but let's start with ribulose 5-phosphate. So here's your 5-carbon sugar. It's a 5-phosphate, so it's 5-carbon. You invest in ATP. Now that ATP came from the light reactions. And using it, we can now have ribulose 1,5 bisphosphate. That is the substrate for Rubisco, ribulose bisphosphate carboxylase. In comes the carbon dioxide. Rubisco does its magic, and now we have 2 molecules of 3-phosphoglycerate. These are just the phosphoglycerates. They're not yet 2,3-phosphoglycerates. I'm not quite sure how that 2 got in there, but these are obviously just 3-phosphoglycerates. The 3-phosphoglycerates are then going to be a substrate for phosphoglycerate kinase, which should sound familiar, right, from glycolysis. And we invest in ATP from the light reactions. Now we have bisphosphoglycerate, 1,3 bisphosphoglycerate. And it is, again, the substrate for glyceraldehyde 3-phosphate dehydrogenase, but with a difference. This one is not identical to the one we saw in glycolysis. This one uses NADPH. And it's going to, the NADPH reducing power is going to be used to reduce this to glyceraldehyde 3-phosphate, and it's going to throw away the inorganic, it's going to throw away an inorganic phosphate which came from this end of the bisphosphoglycerate. So this is a fairly high energy molecule, and the implicit energy or the potential energy in this phosphate is what helps to drive the reaction in that direction. And so now we have a glyceraldehyde 3-phosphate. So this is similar to the reaction in glycolysis, but it uses the NADPH because this is a catabolic, purely a catabolic, anabolic, purely an anabolic process. And then what? So the glyceraldehyde 3-phosphate then becomes part of a series of reactions that allow, oh, that was exciting. Let me try this again. Here we go. Becomes part of a series of reactions that allow, and of course I've lost my whoozy. There we go. What are your options? Arrow. Options. Okay. So there's going to be a series of reactions that allow us to rearrange the carbon backbones, both to regenerate 5 carbon backbones, so that Rubisco has substrate again, and to be drawn off to make glucose and amino acids. If you want to do a very crude look at the bookkeeping, if you have 3 molecules of ribulose 1,5-bisphosphate and 3 molecules of carbon dioxide, you'll make 6 3-carbon pieces, because each time you take a carbon dioxide to one of the 5 carbons, you're going to break it into 2. And these 6 all together can be rearranged back into 3 5-carbons, if you think about it, plus 1 3-carbon that's left to be used in various ways. 3 times 5 is 15 carbons. We're throwing in 3 carbons with those. Here we have 18 carbons altogether in the form of 6 3-carbon fragments. Rearrange, and that little arrow there is standing for a great many reactions. We're back to where we began here with 3 new carbon atoms that came out of the atmosphere that can be used for synthesis. And we're going to take a little look at these reactions to see how those reactions resemble the pentose phosphate pathway. I think I'll be showing that slide in just a little bit, actually. Yeah, in just a little bit. This is obviously a very important pathway, critical pathway. And so any pathway that's that critical is probably going to be regulated. And it certainly is in this case. One of the best ways of doing that is by coupling the Calvin cycle to the light reactions. Since the Calvin cycle absolutely depends on the products of the light reactions, which are ATP and NADPH, coupling those two things would be a useful way of going about it. One part of it is that Rubisco itself has an allosteric site that can interact with carbon dioxide. And it looks a little bit like this. So there's a lysine in which the carbon dioxide can actually become covalently attached if it's at a high pH. And since during the light reactions are pulling protons out of the stroma, the stroma is going to have its pH rise as those protons go away. And so the higher the pH, the more activated Rubisco becomes. It also releases a proton in the process, so it tends to buffer itself back down. Another component that regulates it is magnesium, and the magnesium will also tend to enter the stroma as the light reactions go on. Many of the other enzymes are going to be regulated through ferredoxin. You remember that ferredoxin was the electron acceptor for photosystem 1, and that the ferredoxin was either going to be used to make NADPH or to go back into cyclic electron flow to just make more ATP. But it implies that we're going to have more reduced ferredoxin around when the reactions are more active. And this gives you a feeling for how many of the pathways, how many of the enzymes are regulated by thioredoxin. We mentioned Rubisco. Fructose 1,6-bisphosphatase, we know from gluconeogenesis. That's the one that takes off one of the phosphates through the midpoint. Glyceraldehyde 3-phosphate dehydrogenase. It's part of a number of different pathways. Acetoheptulose bisphosphatase, you may remember that one's name from the pentose phosphate pathway because it's one of the very few sugars that has 7 carbons. Glucose 6-phosphate dehydrogenase, that is in fact one of the key enzymes in the pentose phosphate pathway. One we haven't seen before is phenylalanine ammonia lyase, and this is part of lignin. Lignin is one of the more complex components of plant cell walls, and so if you're doing active photosynthesis, you're probably trying to grow, and if you're a plant you're trying to grow, you need more wall. And so by regulating the activity of a wall synthesis enzyme through photosynthesis, a product of light reactions and photosynthesis, you couple the two. Ribulose 5-phosphate kinase is the one we talked about that will make a 1,5-bisphosphate. And NADP malate dehydrogenase is not so obvious yet. It's part of what we call the C4 pathway, and we'll see in a little bit what the C4 pathway has to do with all of that. But it's another anabolic pathway. I'm including this picture because I think it is at once one of the clearer diagrams of what happens in the Calvin cycle, and also perhaps one of the most beautiful diagrams I've ever seen for metabolism. And I'm going to go through it a little bit to help you get a sense, I hope, of what goes on in the dark reactions of photosynthesis. These are all happening again in the stroma of the chloroplast. So here, let's look at what we're going to have to invest from the light reactions. We're going to be putting in ATP and carbon dioxide, ATP again, and NADPH. So if we start off with 3 molecules of ribulose 5-phosphate, and so you can see 3 arrows there to show that you've got 3 molecules of it. We need 3 ATP, and now we have 3 molecules of ribulose 1,5-bisphosphate. Again, 3 arrows. Those are the target for Rubisco, and they're going to receive 3 carbon dioxides. And Rubisco is going to use that to add and then split the molecule. And now we have 6 molecules of 3-phosphoglycerate. Now we invest 6 ATP. And now we have 6 molecules of 1,3-bisphosphoglycerate. Again, this should sound familiar from gluconeogenesis. That then will be the substrate for the next reaction, which will use the NADPH, the anabolic form. And now we have 6 molecules of glyceraldehyde 3-phosphate. One of those can come off as DHAP, and that DHAP can be used for anything. It can be used to make sugar, it can be used to make amino acids or whatever. The other 5 are going to have to be recycled to get back up to the 3 molecules to the 5-carbon backbone. I'm not asking you to memorize what's going to happen in here, but let's take a little look at the sort of things that go on. So 2 of those 3 carbon fragments can give us a fructose 1,6-bisphosphate. And you throw away a phosphate, now you've got a fructose 6-phosphate. Take another 3 carbon fragment, and now we can use that to make a 4 and a 5 carbon. Take another 3 carbon fragment, and we can take the 3 and a 4 to make a 7 carbon fragment. Take the last of the 3 carbon fragments, with a 3 and the 7 gives us a 5 and a 5. And we can rearrange that 5 into ribulose 5-phosphate. So here we've got 2 of the 3 we need back. What happened down here? Well, we had the 6 carbon and the 3 carbon, which rearranged into a 4 carbon and a 5 carbon, which got rearranged into ribulose 5-phosphate. So it's actually fairly straightforward. It's really not that weird. You have to throw away another phosphate here as you get back to there, because obviously at this point you only have 3 phosphates on the backbones. So you threw away 1 there, you threw away 1 there. And all this kind of rearranging that's going on is the same kinds of transketolases and transaldolases that we saw in the pentose phosphate pathway. I hope you like this diagram. I thought it was the most intelligible form of the Calvin cycle I've ever seen. So, okay. Everything we've talked about so far is usually called the C3 pathways because the product of it makes a 3-carbon fragment. And certainly it's an important aspect of photosynthesis. All plants can do the reactions we've talked about so far. All plants. Some plants have, in addition to those pathways, other pathways that are called C4 pathways that are specializations so that plants can go on fixing carbon when it's hot or when it's dry. And they're called that because the first product of carbon fixation is a 4-carbon molecule instead of a 3-carbon molecule. One of the cute things about these pathways is they can literally happen in the dark. We talked about the Calvin cycle as being part of the dark reactions, but usually they're happening in light. They just don't have to have light around. The C4 pathways, some of them actually are in the dark, and then their products are used when the light comes back on. The C4 pathways are not cheap. They all involve investing additional ATP and NADPH over and above what the Calvin cycle required. So C4 photosynthesis is always more expensive than ordinary C3 photosynthesis. But we're going to see that it's worthwhile for several reasons. In some cases, the two processes are separated in time, light versus dark. Sometimes they're separated in space, and we'll see some of that. But you'll certainly recognize how powerful it can be because both grass and corn are among the conspicuous examples of C4 metabolisms. That corn makes Iowa rich because it has C4 photosynthesis. Okay. So let's take a little look at what's going on here. In the C4 reactions, you have a 3-carbon fragment, phosphoenolpyruvate, which we recognize from glycolysis. And we can fix carbon onto that through a carbonic anhydrase that produces bicarbonate and the enzyme phosphoenolpyruvate carboxylase, which actually should be familiar to you from before. And now we have the 4-carbon molecule oxaloacetate, dicarboxylic acid. Oxaloacetate, of course, is also familiar to us from the citric acid cycle. It's one of the intermediates of the citric acid cycle. The oxaloacetate is then reduced to malate through an enzyme that's given its backwards name, malate dehydrogenase. And in this case, we're using NADPH. Similar reaction occurs in the citric acid cycle, but there in the mitochondrion it uses NAD plus or NADH, depending on which direction it's going. So now we have malate, which is the somewhat more reduced form. You can see here is 2 bonds to an oxygen. Here's 1 bond to an oxygen. So this is the carbon that got reduced. And we invest, we can then recover this carbon dioxide from that end through an enzyme called malic enzyme, which will split off that carbon dioxide by another reduction reaction and restoring to us now pyruvate. So it cost us a high energy bond to do this, and it cost us a recoverable high energy electron. This carbon dioxide is now available to use in the ordinary Calvin reactions. For the C4 reactions to keep going, we have to get the pyruvate back up to phosphoenolpyruvate. And those are the usual reactions that we've seen before. That costs us both ATP and GTP to get back to a phosphoenolpyruvate. So the overall cost of this is fairly substantial. But what it does allow us to do is to capture carbon dioxide for the Calvin cycle. And the only advantage to having, okay, here's one more of the other reactions here. This is where part of the cost comes in, that you're throwing away your inorganic phosphate when you do the phosphoenolpyruvate carboxylase. This is the opposite reaction of what we saw in gluconeogenesis. In gluconeogenesis, we took an oxaloacetate and made a phosphoenolpyruvate by spending a GTP. In this case, we want to build rather than to break down, but we're still spending a high energy bond because we have to throw away the inorganic phosphate. And we talked a little bit about this being in the opposite direction from the citric acid cycle and using the NADPH. And then there's the malic enzyme. It's similar to a shuttle enzyme we saw earlier, but again, this is using the NADP plus. And so on. So now we do have carbon dioxide available, and what are we going to do with that? The reason this works, or the reason that this is worth doing at all, is it allows the plants to pick up carbon dioxide with a higher affinity than Rubisco is capable of. Rubisco is not a terribly good enzyme. It's not a terribly high affinity for carbon dioxide. It's not very smart. It sometimes picks up oxygen instead of carbon dioxide. And so what happens in a C4 plant, for instance, is something like this. You can, in the mesophyll cells of the leaf, the carbon dioxide comes in. It's fixed into an oxaloacetate, reduced to a malate. The malate is transported from the mesophyll to another cell called the bundle sheath cell. And the bundle sheath cells are specialized in these plants for this process. It undergoes the reaction that releases the carbon dioxide in the chloroplast, and that allows the Calvin cycle to run there. The pyruvate then has to be returned to the mesophyll cells, and has to be regenerated back into phospholipopyruvate at the cost of high energy bonds. So overall, it's going to cost us 2 high energy bonds for every carbon dioxide that we're going to fix. But because these reactions have a higher affinity for carbon dioxide, and because they don't make a mistake between oxygen and CO2, it allows it to be able to operate more selectively than the standard version of the Calvin cycle. It also allows it to separate it in time. So for instance, you can do it in the Crassulaceans. They do it at nighttime. They do the first steps at nighttime. When it's relatively cool, they open up their pores and they take in the carbon dioxide and they make a bunch of oxaloacetate. And then when the lights come on, they turn it into the malate and finish up the rest of photosynthesis. In things like corn, it's separated in space. And so the bundle sheath cells are hardly exposed to the atmosphere at all. They're going to get their carbon dioxide through this process, but they're not exposed to the oxygen significantly. And so they have much lower rates of photorespiration. Since photorespiration also costs energy, it's not quite as expensive as it might look. So what are we going to do with the glucose that we make through the Calvin cycle? Well, we're going to make carbohydrates. And this should look rather familiar to you because there are some similarities between this and glycogen synthesis. So here's the monomer, glucose 1-phosphate. And glucose 1-phosphate by itself cannot be used to make into polymers. We mentioned back several weeks ago the idea of an activated monomer. And in this case, we activate this monomer with adenosine. So we end up with ADP glucose. So you have the glucose here with its phosphate, the adenosine and one of its phosphates. The 2 other phosphates from the adenosine are now inorganic pyrophosphate. And that is split into 2 inorganic phosphates, and that helps to drive the reaction. The ADP glucose is the activated monomer that is now a substrate for starch synthesis, and starch synthase can use that to add another molecule onto the end of the linear polymer of starch. This particular reaction using ADP glucose is present in bacteria as well as in plants. So it's not just a trick that plants use to do it. If you add this all up, what it's going to end up costing is 1 high energy bond for each glucose that you store this way, just as it was for glycogen, in fact. Another molecule that cells use quite a lot is sucrose. Sucrose is a disaccharide. Here you can see the final product of the disaccharide. And it's a dimer, it's a disaccharide composed of a glucose and a fructose. So here we have a fructose 6-phosphate, which we could get from the Calvin cycle. And here we have a UDP glucose in this case, not ADP glucose. And the UDP glucose is the activated substrate that's going to fuel making the bond between the 2 of them. And so now you have an alpha 1,2 linkage between the 2 molecules. Now what's interesting about this alpha 1,2 linkage here, in contrast to some other disaccharides, is that there are no free aldehydes exposed to the rest of the environment. So sucrose is not an oxidizing sugar. It is rare for sucrose by itself to be added to other things. And it's possible that that's why plants like to use sucrose as their preferred transport sugar, or for things like sugar beets or for sugarcane, even as a storage sugar. But it is less reactive. A diabetic, for instance, who has high circulating levels of glucose in their blood, you can detect proteins in the body that have had glucose added to them non-enzymatically, because glucose as a molecule is slightly reactive. Sucrose, on the other hand, is not. Unfortunately, we haven't learned how to use sucrose in our bloodstreams. The other thing we can use the UDP glucose for is to make cellulose, which is, again, a linear and branched polymer of glucose. Starch and cellulose are both polymers of glucose, but they differ in their linkages, right? Okay. It is not just plants that engage in photochemistry of one kind or another. There's some photochemistry in animals that's quite important. And let me just touch on some of those. And so, for example, as you might think, most animals have some kind of molecule that can detect light. Almost every non-plant has an ability to entrain with the light cycle from day to night. They generate what's called a circadian rhythm. And circadian comes from circadian. That is, a circadian rhythm is around a day long. The endogenous rate of a circadian rhythm will be close to, but not exactly 24 hours. So if you lock one of us away in a room with lights on all the time, our body functions will tend to cycle at around the 24-hour point, but apparently humans came from Mars because we tend to cycle at about 24 and a half or 25 hours, which is closer to the day length of Mars. But if you want to stay on cycle, being re-exposed to light every day will then re-entrain that circadian rhythm. This is one of the reasons that human beings need real daylight. These lights, they're not strong enough. Real daylight is what's needed. And these were called cryptochromes for a long time because the sensor was not known. It turns out to probably involve both flavins and pterins. And if you look at the structure of the flavin here, you can see it has the repeating aromatic bonds that are so characteristic of molecules that can absorb light. Its peak absorption is around 450, so it's sort of more near the middle of the range. It is, in fact, related to molecules such as FAD or FMN. It's the flavin part of flavin mononucleotide or flavin adenine dinucleotide. But in this case, it's valuable because of its ability to absorb light rather than because of its ability to undergo redox reactions. Here's the pterin molecule. Pterins, again, have this kind of aromatic resonance that looks like a candidate for capturing things. It acts as an antenna, so it can capture light and transfer that excitation to the flavin, the same way we saw carotenoids capturing excitation and transferring it to the chlorophylls in the reaction centers. Are there other questions about this before I go back and take a look at some other diagrams of photosynthesis that I thought might help bring out some of the similarities? Or are there other questions? Yes, they do. That's an excellent question. Plants do indeed have mitochondria, partly because the sun isn't on all the time. And so at nighttime, that's the only way they get their energy. But also because the ATP that the chloroplasts make stays in the chloroplast. What happens in the chloroplast stays in the chloroplast, you know? And it's the so to speak. The chloroplast can export glucose, and they do, but they can't export ATP. And so the plant cells definitely need mitochondria in order to make ATP for the rest of their functions. No, photorespiration is due to this side reaction of the rubisco. Let me just show you that again, because it is a little confusing. And let me get back there. That would have been slide number 24? Okay. And this is worth having straight, because you're quite right, it has the potential to be very confusing. Wait. Come on. Um, it's a little unfortunate that the words sound so close, respiration and photorespiration, because it sounds as though they ought to be either the same thing or similar things. What's common to the idea of respiration is that oxygen is consumed. And so in ordinary respiration, oxygen is consumed and ATP is made. And that's what happens in the mitochondria. Photorespiration is only in the chloroplasts, and it's because of this side reaction from Rubisco that oxygen can take up the place of carbon dioxide. In that case, you only get a 2-carbon fragment and a 3-carbon fragment. And it's a side reaction. It's not that there's such a lot of similarity, but there's so much more oxygen than carbon dioxide. Yeah? No, I don't think I did. I was trying to make a contrast from that. If I made that confusing, I'm sorry if I did. Respiration is what happens in the mitochondria. It's a good thing. It's when you consume oxygen to burn carbohydrates or to burn fats. And plants and animals can do respiration. Photorespiration is this side reaction that's undesirable. It's one of the reasons that C4 pathways exist, is to avoid photorespiration. So there's good respiration, and then there's photorespiration. Respiration itself simply means consuming oxygen. And you can consume oxygen and have something to show for it, or you can consume oxygen because you made a mistake. Is that clear now? Is that clear? Okay. Are there other questions? Okay. I found some pictures that I think will make the parallels between the behavior of the chloroplast and the behavior of the mitochondrion a little clearer. So if you'll bear with me, I have some new pictures. I don't have a handout for you yet, but I will put these on the web so everybody can see them. And what I like about these pictures is that they are color-coded in a way that I think helps clarify some of these relationships. And I'm sorry I don't have a handout, but since it would be in black and white anyway, it would be of only partial use. And how can it take this long to mount a picture? Come on. Come on. Okay. All right, let's try this one. Okay. So take a little look at this picture. What this picture has captured for us is, in the top part of it, it has captured the spatial physical objects. And so you can see here, I wish there's some way to make this the default. Oh well. So you can see here in the physical orientation of things, photosystem 1 as an integral membrane protein that has protons moved and generates oxygen. You can see the plastoquinones in the membrane. They're lipid soluble, carrying electrons to cytochrome B6F. B6F is itself an integral membrane protein, moving protons and making plastocyanin, the peripheral membrane protein. Plastocyanin transfers that to photosystem 2. More light energy there. Now we can transfer electrons to ferredoxin, and in the linear pathway, ultimately to NAD+ to make NADH. All of those reactions, this reaction and this reaction are both of them moving protons, creating a proton gradient in the lumen of the core of the crystal. Those protons flow back down through the ATPase and make ATP in the stroma. Okay. Now, what's handy, I think, is this one actually color codes it then to the electron motive force, the electron potential of these things, showing you the change in potential in photosystem 2, that it's a downhill to plastoquinone, downhill to cytochrome B6F, downhill to plastocyanin, and then a second boost to re-energize the electrons to go to ferredoxin and ultimately to NADPH. So this one color codes those relationships in a way that you can turn the physical objects and relate them to the energetics of them. And then the next diagram uses the same colors to try to give you a parallel to the mitochondrion. So at the beginning of the transfer chain for complex 1, and so forth are colored in the same sequence. You again have ubiquinone now as the lipid-soluble carrier. You have complex 3, and which also can do the same thing, but then you get to, sorry, this should be complex 2 here. Then complex, where are we here? Complex 3, I'm saying. So complex 3 is giving you the cytochrome C. It's the peripheral component in this system, transferring it to complex 4, which is ultimately going to use oxygen as the acceptor here. And again, there's protons being moved. Now we face the little delicacy that for some reason mitochondrial people and chloroplast people think in opposite directions, okay? Here is the storage space for the protons in this diagram. The intermembrane space would be comparable to the lumen of the crista. Here's the matrix for this diagram. That's comparable to the stroma. So in this case, the protons are going to flow downhill and make ATP. If you were to flip those two diagrams, you could see that the protons are always moving out of the jelly interior across a membrane into a topologically exterior space. And they're always flowing back to make ATP in that jelly interior. Okay? I'll offer you this as well if you want. This has a text to go along with it. It's for people who think with their hands as well as other things. It's a chance, it's a fill in the colors for ATP synthesis. And I know I learned most of my anatomy with the anatomy coloring book. So if you find this kind of thing handy, you're certainly welcome to use it. It'll be on the web. So, and I think that'll be it for today. Please have a wonderful weekend. Thank you. I'm happy to answer questions if people have any more. It's what? There is, and I'll include that as well.